All right. Well, welcome back to Engineering 443, 543 on computational biology modeling methods, virtual tissues. I need to remind you that, as usual, this is being live streamed on YouTube and recorded, so please be aware of that. Today, we're going to move forward a little bit to thinking about concentration fields and diffusion. Before we do that, I would like to review briefly some of the things that we started talking about last week and were in the homework, but that were not talked about so much in class. And we can either go over them as sort of lecture form, or we can, if somebody's got an answer that they have in their homework, they could show it and show the class how to do it. So either way. I'd like to start out by getting some quick student project updates. Remind you of a few things that we did last time that I think are relevant. Then we maybe talk a little bit. I don't think we'll do all of those exercises on contact inhibition, but we might do one of them together. And then start talking about concentration fields, transport, and the diffusion equation. And that diffusion will take us through today and probably next week. And also begin to talk about things like, this week we won't start talking about chemotaxis, but we'll start talking about that next week. And I know some of you are working on chemotaxis, so maybe you can do the lecture for me. So why don't we start out very quickly with the project updates, if there's anything that people would like to report. Please let me know, and just go around the room and get a quick update. Start with Joel, I guess, since you unmuted first.
Okay, yeah. Me and Jackson, we are working on this project together. We changed the project now. Our idea is to do different types of cell death. So we're thinking on getting a bunch of epithelial cells and basically work on how they will die exposed to a different pathologies. Or the idea is having a specific virus and a specific bacteria and a specific fungi maybe, and at least one immune cell to signal the death that we see on the immune system. The whole purpose of our project is thinking of building this module that can be plugged in different projects in the future. So it's the stepping stone of a cell response. That's our goal here.
Okay, that's ambitious, but also very valuable. You have Claire Bryant's work on pathways. There's the work by the team at St. Andrews University on modeling that, and we should look at others. But I think that's fascinating, and how cell death is mediated. It's a big open set of questions. How inflammation is mediated in tissues through cell death. It's a critical question. So that would be.
Our hope is actually to make something that can be used in the future. So the particular goal is to make things work little by little and see how far we can get in this project. If it's something that can be expanded in the future, we're glad to keep working on it too.
Great. Okay, Connor.
So Andrew and I started the first week of our ambitious get ahead of things plan. At first, it seemed like we made it most of the way through it, and we had the code running and working. But then today, when I went to run the code to get screenshots, it crashed Compusal, and Andrew had the same issue. So we are now in the midst of figuring out what's wrong and why it worked before and does not work now, which will be an interesting point. So we have two things that are partially in progress, which we might email you about to kind of show where we got and where things are at. Besides that, quick clarifying question, because it's possible I misread it, but on Canvas and on the project sheet, it said that we were supposed to give 30 to 40-minute presentations today.
Today? No.
I think
Did
I
I?
could have sworn that it
I
did.
may have gotten the timing wrong. I was expecting reports last week. So maybe
No.
I got the dates wrong.
He's right.
Okay.
It's due. It says it's due today. Our 30 to 40-minute paper presentation.
Do people want to do that? I don't see a huge groundswell of enthusiasm.
I mean, Andrew and I did it, but we are prepared to wait for the actual date. I just wanted to clarify.
So I guess here's how I'd put it. It doesn't have to be that long. On the other hand, I think organizing your materials is important. Now, for example, if Joel and Jackson have just changed topic, it's not reasonable to ask them to be ready to do a 30-minute presentation on their new topic when they've just changed topic. And if we had 6 people all present 30-minute talks, it would chew up our whole class. So my hope was that maybe possibly starting this week, or we could start next week. We don't want to wait too long, because if we wait too long, we're already at the end of the semester and then it's not doing its job. But if we say start next week and we do 1 or 2 presentations that are 15 or 20 minutes long, and then the following week we do the rest, then we're not only doing student presentations for all class. It's a little bit of variety. And so I was hoping essentially that we would have people ready to queue up so that we could get started. And in my experience, when you give assignments like that, there's some people who aren't ready. And I'm not saying that's the fault. Sometimes that life happens. But the idea was to try to get started. If there's no deadline, nobody does it. So if you'd like to present today for 20 minutes, the floor is yours. And of course, if people are partnering, then we have fewer total talks to give, which also reduces the total amount of screen time we're using. So it's up to you. If you want to present this week, that's fine. If you'd rather present next week, we could do that. Your choice. But I do want everybody, and 30 minutes, I'm not sure where I came up with the number 30 minutes. I want people to feel they have enough time to present in detail. On the other hand, if people talk if 15 minutes it's like asking me how long should the paper be. I realize it's much easier if I say 10 pages or 12 pages. To say long enough is hard unless you have an example in front of you. But it's a little bit with the proposal. People have presented slides already of things. And so now the question is, can you work it up and build a more formal proposal? I'm asking people to do these self-assignments. I'm asking people to start diagramming their ideas. And so depending on how far along you are, you might present more background material, more work that you've done, more plans. I think coming in and doing one slide saying I haven't decided is probably not answering the question. Coming in and talking for 3 hours could be fascinating, but would be overdoing it. And so sort of the geometric mean between those two things is 30 minutes. So that's how I came up with the number. I'd say enough slides that people, not just I understand what you're doing, but your classmates understand. The background, why you're doing it, what has to be done, what the challenges are, how you think you're going to do it. By the time you do that, it takes 20, 25 minutes. And in my experience, the shorter the talk, the harder it is to write. because when you're going to give a very short talk, you have to be ruthless about what it is you select. And that's actually quite, I mean, some people, I guess it's easy for, but I find it difficult. It's easier to give a longer talk because putting in all the things you think are interesting is easy. Trying to decide what are the essential things is actually a lot of work. And so I wanted people to feel that they could use that time if they needed it. Does that help? So why don't we say next week? And I apologize for the time. I think this was assignment 4A, wasn't it? That we're talking about, yeah. So again, the way the timing was done is I wanted people to be ready, and so that if but it wasn't that you had to present today. Again, if somebody would like to present today, you're welcome. I'm happy to yield the floor to you.
Really quick, on Canvas, it says homework 4A is our mini project plan. So I thought that was like our, roadmap, but you have a different one specifically called in-class presentations, and it's due today on Canvas.
Okay, so I saw those as partnered assignments. And probably what happened was I think I set up all of those initial assignments at the very beginning of the semester when I set the whole project. And the reason I tried to do that was because the last semester people got sideswiped, where the very first assignment I gave, it said, by the way, you have to do a report, a 10 to 15 page report. And at the end of the semester, people said, what report? Because by the end of the semester, people had forgotten that the original assignment had that in it. And so my goal here was to try to keep people remembering stages of the project. And if there's an inconsistency and you send it to me, I'll try to reconcile the inconsistency. So again, Josh, if you want to present today, you are more than welcome to do it. I would be delighted. But if you would prefer to have another week to get organized about it, that's fine too. And again, there's an issue with Canvas, which is when you do uploads, if you want to upload more than one thing per assignment, you have to zip it. You can only upload one object per assignment. And so if I think you're going to give me a slide deck and a write-up, I will sometimes create a pair of assignments so that there's a placeholder for both of them. And I understand that can be confusing, and I appreciate you pointing it out. Again, your patience is much appreciated, and when you point out mistakes, I will try to fix them. So does it? Sorry, Connor. So let's go back to Connor, and then we'll go to you next, Josh. So Connor, let's just keep.
So actually, Giuliano, you're the best. Thank you. That is exactly what happened. We were playing around with different types of diffusion solvers for the chemical fields, and when we switched out the one for the other, that Compusal was not happy with it. I'll probably need to talk to you more about why it's not happy with it to understand. But so we do have progress for this week. We got it so we could measure the velocity as it's chemotaxing and get it to switch directions based off of its position on the Cartesian plane. And then where we ended up making some progress but got stuck was setting up a chemical field with holes in it, like they did in the original paper. But we made some progress with that. So that is officially where we're at.
If you want to show, go ahead.
Oh, the issue that I came into was I was only able to get the chemical field to display when it's initialized, and then when it goes into the step function, all the pixels that have a gradient just immediately switch to NA values. But otherwise, we have there's a focal point and then the chemical gradient both decreases in like how much gradient there is, and then the probability decreases linearly, like just a Bernoulli, coin flip. Like a weighted coin flip, it decreases, the chance of it happening decreases over time.
That's a great way to do it. I mean, so today's lecture will be basically review for you because the other people in the class haven't learned about fields. So maybe you maybe should be giving the lecture. If you want to show your results, I can certainly give you the screen share if you want to show us what you got.
Yeah, I can give a small display of it. Yeah,
that would be great. I know there was one. I think somebody had an issue with a crash of player and for plotting, and the issue that came up was one thing that can be confusing about Compusal. It occasionally tries to be friendly in terms of error messages, but it isn't always. And Giuliano, maybe wrapping things in a try would be helpful, which is if you put the window initialization for scientific plots in the init instead of in the start function, it crashes player. And it'll throw an error briefly and then player will disappear. And it's always hard to debug things if player disappears, you don't see what you got. And so that's one that's tricky. The same kind of thing can happen with tracking fields. Tracking fields, the initialization of the tracking field has to be in the init, but it has to be after the command inside of the init that says a dot init. If you put it above that, it will also cause strange player to disappear on you. And so maybe Compusal should have a try function there so that at least when it dies with an informative error message rather than crashing player. So those kind of things definitely happen. And the best thing that I can say is that if when we're made aware of it, we'll put it on the stack of things to fix. Another thing, Giuliano, you might show people how to do is to set up movies, to take time series movies and screenshots, because we haven't really gone over that, and we're getting to the point where that would be useful. Okay, Andrew, please go ahead.
So what here is that this only displays during the start function, and as mentioned, like whenever there's a chemical gradient, it goes to NA. But what actually is like how I implemented it is I have you just select a point in the lattice, and that is where the most gradient will be, most of the chemical will be, and then you're basically calculating the focal point to the most distant point, which it will be one of the corners, and then basically for every pixel, the probability of the gradient being there decreases. I just put it's a NumPy function. It's I put binomial with one trial, and then the probability is based off of the Euclidean distance, and then also that distance scales how much of the gradient is there. What's actually done in the paper is a combination of this plus some noise, and so I can actually I've been thinking about adding in some sort of noise as well.
Great. Well, I wonder why it's disappearing on you. Maybe when we go forward with the exercises, since you'll have already done some of them, you could play with that and then show us what you've got, and we can try to do some debugging, or you and Giuliano could go into a breakout room and try to do some debugging as we go forward. But that's great if you could create an initial condition like that. That's already a lot. I never tried to create noisy gradient fields. That's an interesting thing to do.
And this is just a good way to make it automatic, because we have seasons in our simulation. So we're just going to initialize another random point in the lattice and then apply the same thing.
Yeah, it makes sense. Then you have a function that throws the chemical down the way you want. Excellent idea. Okay, anything else, Connor and Andrew? I'm delighted you've got the chemotaxis working. That's great.
Yeah,
Look forward to seeing it.
do you want me, I know you said that the chemotaxis file that we were debugging a couple weeks ago, that you had a working version of it, but it vanished. Do you want me to send you the base version we have now that does the flipping at the sides of the walls so you have it again?
That would be great, and I suspect your classmates would appreciate having it too, because as we start doing chemotaxis, they'll need it, and I think it'll be useful to have. That would be wonderful.
Okay.
A little hive mind, collective sort of crowdsourced programming for that.
That's how the best science gets done anyway.
Okay. So let's see, we have Alex.
Yeah, so I've been making good progress actually, although it doesn't seem like it on the initial. I've managed to get CC3D building on both Linux and the Windows machine, which that took a lot longer because Windows is not fun to work with sometimes. The CC3 build setup is very interesting, and like you need like older, because uses like older versions of C++ compiling stuff. So bringing it up to the latest version took a while. But then with all that working, I've been able to convert one of the functions in the field extractor to be parallelizable, such that before it would iteratively go over the cells, and if it found one that worked, it would add it to it like a data set. Now, to make it be parallelizable, you have to, sorry, I can't. Let's get rid of her now. You have to pre-generate the whole data structure you want to put data in, so then you can insert things correct into the right locations. And I've gotten that working, although it is slower. At least, I guess it depends on what you're looking at, because before, if it was mostly an empty area, it could skip all of those. Now you have to include those empty points. Like technically on a full like 2D field, it would be equally speedy. So it's strictly slower, but not always slower. But now with that working, I can try and integrate OpenMP into that function, and then start working on all the other functions.
Well, that's interesting. I suppose you could basically inquire of your environment and say if the environment is multi-threaded, then you use the slower but parallel version, and if it's not, then you use the faster one that's serial.
Yeah, that would be an option. So I'd have to talk with, I don't know who like maintains the code more. But the downside of that is you now have two code paths you have to support. So you're doubling the size of your code base, at least in this small instance, but so that it may not be desirable for that reason.
I'm speaking out of turn. I'm not the software engineer.
I mean, it's a worthwhile idea. It's just a matter of is it worth it?
I guess the kind of place that you wind up having to do that is something like when you're doing GPU acceleration, and you have to have a CPU version,
For sure,
because not everybody has a GPU to run on. I guess the nice thing with the OpenMP is that if it's one thread, as long as it works for one thread, then you don't care how. I mean, you won't get any acceleration, but you're not losing anything. You won't crash because of it. So I think, I mean, if whatever you and TJ decide is fine. And maybe patch Maciek into it if we need to.
Yeah. Now that I have one of the harder parts was figuring out how to do all the stuff in VTK. because it's not exactly obvious, because there's a bunch of different data structures and you interact with them in different ways. But it's all been, so now that I know most of the data structures used in the field extractor are the same, so it should be a lot easier to work with the other functions, which is good.
I think this is great. I mean, I know, not here, but TJ was showing off that he's got a version of Compusal basically that now runs inside of Jupyter Notebooks.
Yeah, he showed me that too.
And so that's also an exciting idea that you'd be able to run it as a client inside of Jupyter Notebook. I mean, Player, remember, Jupyter Notebooks is a much younger project than Compusal. So Player was a great idea when we did it, which was a long time ago, because there wasn't anything else. But now it's not obvious that having all that functionality. I think this is great. I think it'd be really useful. I appreciate your doing it.
Yeah.
Okay, so I guess who's left? Josh, you've been very patient.
So I was planning on giving the paper talk that I thought was due today, but if that is not the case, then I think I would like to wait until next week. But I mean, I read the paper and I feel like I understand a lot of what he was doing. He went over, all of he didn't really go into too much detail about the actual model, but he talked about, how he set up chemical fields and they had secretion, diffusion, and decay and all that. But I think I'll just work on my presentation a little bit more. And also, I noticed that there is some pretty useful information in the comments of his code. So I think I've looked at his code, but I haven't looked at all of them. And so I think I'll go through them and read the comments. And I found out he was using specifically Compusal version 3.2.5. So I might try and download that from the binary archive and run it. And then once I get that working out, I'll try and update it to 4.2.5.
Wow, that's old.
Yeah.
There was the Environmental Protection Agency uses our code, and in many situations in the history of computing, maybe not so much these days, people will freeze on a code version. Used to be that with operating systems, they get better and better and better, and then they get worse and worse and worse after a while. And so people would say, okay, I won't do any updates after a certain amount. So a lot of people like Compusal 3.7. And then 4 was not backward compatible. And there were that lack of backward compatibility bought us a lot of things. They're a lot cleaner in many ways. But boy, when you have a government agency using Compusal, they really did not like having to refactor their code out of 3.7. So they only moved, actually just in the past week, they finally moved from 3.7 to 4. So there'll be issues. Structurally, the models should work. It's possible there's something strange in there that was in an old version that got lost. But mainly what'll change is the boilerplate at the top. All those are the classic Python boilerplate that you ignore will be different. And again, I'm happy to walk with you. Giuliano is very good at doing those updates too. So definitely schedule a meeting with Giuliano. And if you get stuck with TJ, and I can work with you as well on that. But that's fine. And also on these presentations, do give it a good effort. But don't kill yourself. It's not a final presentation. It doesn't have to be terribly polished. If there's a typo in your slides, don't worry about it. On the other hand, do try to get the ideas across clearly. Oh, by the way, Giuliano Thursday is giving a seminar on his Compusal models. And so if anybody is available at 3 o'clock on Thursday afternoon to attend Giuliano's seminar, virtual seminar, and support him, I'm sure he would be extremely grateful. And you could perhaps look at that and what you like about the presentation and how to present these ideas. Would be good to help you think about your own things. And what you don't like about it, you can give him advice for the next time he gives a talk. Or you can say, gee, the way he did that isn't clear to me, and that may help you think about how you want to present the material yourself. So, Giuliano, could you put you put the link in the chat already? Yes. So if you click on the link that Giuliano has just put in, it will take you to a page that will have a registration link. And the meeting is open to anybody, but you have to register because we've gotten Zoom bombed a few times. So it's open, but you have to give it your email address to join the meeting. But this is this National Institutes of Health sponsored working group. And as I say, I know people are busy, but if you're willing to come and support Giuliano in his presentation, it would be great. Would mean a lot.
We have a class at that time. Is there going to be a recording of it, or is it the type of thing there will be? Okay.
So if everybody has a class, then everybody has a class. Well, then it's too bad. But it's our working group meets at 3 o'clock to 4:30 on Thursdays. And it's not always modeling, but it's usually multi-scale modeling. And sometimes it's clinicians, sometimes it's experimentalists. But if you want to know about what the world of modeling looks like, it's a good place to come. And again, everyone is welcome. It's a completely open meeting. It's not that often the grad students present there, but it happens. So you're welcome, and the videos are on the same YouTube channel that this class is on. All right, and I guess we still have to set up a meeting with Roland, don't we?
Yeah. So we're just waiting on him in terms of finding the time, and something that I was going to throw out there was that if it ends up that the only meeting time that he's available is obscenely early morning, then Andrew and I could always meet with him and then reconvene with you at a later point that works.
Yeah, I'd get the meeting as soon as possible, and if I can be there, I will. But it's better to do it.
Yeah, no, I agree. I just know he sent an email beforehand saying that his most likely time was 6 to 8 a.m. So I'm fully prepared. He also mentioned like there was a possible 11 a.m. time, but I'm prepared for the possibility that it might be a smaller group early in the morning.
Okay. Well, he's a busy man, I guess. And the time difference doesn't help. But anyway, but I'm glad you're in touch with him. I think that's great. Okay. Anything else before we get started on the lecture part of the class for today? So I guess Josh and Connor, you're on tap to present next week for your presentations, and then the remainder will be given the week after. How's that? Again, with the assignments, since I don't always know how far we're going to get, there always has to be a little bit of fluidity in terms of the assignments. And I appreciate your patience with that. And thank you for drawing my attention to the issue of the scheduling this time. Okay. So let's come back to the lecture. I just wanted to remind you of a couple of things from the last class. Again, the concept of cell type and this relationship between type number, which is called type ID, and type name. And the reason for that is initially, this goes back to the Fortran code that I wrote in 1992. And things, you couldn't index things by name in Fortran. You had to have an array index, and so it had to be an integer. So that's why types are integer numbers, and that's been inherited for 30 years now. And so going from type name to integer is easy. Self.typename in capital letters. You can look up the type name of a cell easily enough, but if you just want to get all of the type names in the system, it's a little bit more complicated to go from type number to type name. And I'll just remind you of that. That's not something that is usually critical, but when you're doing plots, it's nicer to be able to plot the name of the thing rather than the number. And so you may find that useful. The big thing that we did last time was talking about how to do cell division in Compusal. And the key thing about cell division is that when you divide, you have a new cell appear, and you have to decide in particular what pixels or voxels of the old cell get transferred to the new cell. And typically we call the cell that persists the parent cell, and the new cell child cell. And the main idea that you have is a plane of division. And that plane of division can either be fixed, random, or along the long or short orientation of the cell. And you can do fancier things if you want. And then you have to decide how attributes of the cell are inherited. In particular, what happens to the target volume and target surface of the cell. Often we divide by 2, assuming you have equal division, but not always. And then is the child cell of the same or different type? And if you have a model of cell cycle, you might want to ask the question, does this parent stay in the cell cycle or not? And that's not really intrinsic to Compusal because cell cycle isn't implemented in Compusal. That's something you have to add in your Python. The type example comes up because the boilerplate code switches the cell type. And so if you just paste in the default code for cell division, it switches cell type on division. But that self.parent cell, self.child cell is an important concept. And cell division has to be put into a Mitosis Steppable. And typically we do that by setting some condition on the cells and then invoking a divide process and using an update attributes to do the reassignment. One thing you'll run into when you do that is attributes that are put in cell dictionaries are shared between steppables because they're attributes of cells, and the cells are shared. But variables otherwise are not accessible between steppables. And so there is this dictionary self.steppable_shared_variables, which is essentially a place to put globals. And if you prefer to use Python globals, you can do it. But in my experience, it's a recipe for more hard to debug mistakes. Whereas using the dictionary may be a little bit slower for access, but it's reliable. Coming through the Mitosis Steppable, you'll see we typically will create an empty list of cells to divide. We go through all of the cells or some subset of cells in our simulation. We have a condition here, cell.volume greater than 50. We put that cell onto the cell to divide list. Then we go through the list and do the division. And the key question is, why don't you just do if cell volume greater than 50, self.divide_cell, etc. And the reason you don't want to do that is that when you divide a cell, you change the list of cells. And one strict do not in Python is that you are not supposed to change the membership of a list while it's being iterated over. Sometimes you're lucky and Python behaves itself, but it can crash Python completely, or it can give you completely strange, wonky results. There is no defined answer when you change the list you're iterating over. And so, as a matter of best practice, sweep your cells, decide which ones are going to divide, and then do the division. There's the attributes function. In this case, by default, typically we divide the target volume by 2, equal division. And then we do this great clone parent to child, which in principle copies everything about the parent cell to the child cell. And as I mentioned, the default takes the parent cell type and switches it to being the opposite one here. And there are a couple of other possibilities that you can play with. When you're using the Twidit in the wizard mode, it's easy enough. You have a button that creates a Mitosis Steppable. If you already have a simulation and you want to add cell division to it, there is this add steppable function that you can invoke in 2 different ways. And they will automatically then create the placeholder steppable for your division. There are a couple of other kinds of steppable you could add, a generic one, and then this cluster mitosis, and then run before MCS, which I don't think I've ever used, but is available. And then because those visualization fields, remember, go in the init, you can put those visualization fields in here. Most of these things are conveniences, but adding a steppable is something one does. Now, deleting a steppable is something else, because there isn't an automatic way to do that. You have to. If you have an extra steppable, it's better to leave it there, not try to get rid of it. If you need to get rid of a steppable, ask, because you have to do some editing. Okay, so we talked a little bit about regulating cell growth, and we mentioned that in reality, cells change their growth rates for a whole variety of reasons. We're going to focus here on some of the possibilities. And I think in the interest of time, we'll probably skip this exercise and move on to the one with contact inhibition. One of them, and I think this was what you had in the homework a little bit, was if you have constant cell growth, people found that you filled the space and the pressure in the system went up. And maybe if anybody didn't get it, I could ask one of you to show. I know Joel had a nice write-up of this. I think some other people had some good write-ups of this as well. If you keep putting, essentially target volume is like putting material into your system. And you can imagine that material is compressible like gas in a balloon. And so one of the things that you found in the homework was that when you have all the cells growing simultaneously, the cells in the middle of your aggregate were under high pressure. Cells at the surface could grow and divide easily, but the cells in the middle were under pressure. And that's because it takes time for the system to equilibrate mechanically. And effectively, you're dumping material in faster than it can spread out. Once the cells fill the whole lattice, there's no room for that extra material to go, and so the pressure goes up. And so people did a series of simulations, originally due a week ago and now extended to this week on request, looking at how the pressure changes in those situations. And one of the things that I had asked you to do was to compare the case where the cell division happens when the cell reaches its target volume from a situation which happens where the cells divide when the target volume reaches a threshold. In one case, if the system fills up, division stops because none of the cells can ever get to that size. In the other case, the cells will keep dividing, and of course then the target volume will keep going. Each time they divide, initially, when you've divided, you haven't changed anything because you're reassigning target volume by half. But now you have twice as many cells growing at a constant rate, so the target volume is growing twice as fast. So in the case of growth, the increase of target volume, total target volume in the system is growing exponentially as you have cell divisions. And so you get quite a different outcome. When you have cell division based on actual volume, cell division will slow down and stop, although you can get interesting waves of contraction in the system, depending on the growth rate. In the case where the target volume is your trigger for division, the cells will, once you fill the space, the cells will get smaller and smaller and smaller. You'll get more of them. Eventually, you'll get a situation where some of the cells disappear. And then things, again, there can be instabilities. Those instabilities are more physics interesting than biology interesting, but they're there. You get waves of pressure moving through the system. Okay. The other one that I was bringing up, and this was exercise 6.1, was a case where you have a cell type change, and the parent cell stays in cell cycle, but the daughter cell does not, the child cell does not. And in that situation, effectively what you have, if you start with one cell, is that you have one cell that divides, its offspring sit there, it grows and divides, its offspring sit there. So now you have a linear increase in the number of cells with time rather than an exponential one. Eventually, it will also fill the system, and depending on your rules for division, it will either keep getting smaller or stop. But in this case, you get linear growth of the whole tissue rather than exponential growth. And again, I'm not going to, I think we'll skip this detail today for this. The next, well, here's the result. So I've got the picture. So you have here the blue is the dividing cell, red is the differentiated, terminally differentiated cell. It's not dividing. And you can look at what happens to the pressure. I think it, again, I hope as we go on, and maybe next week we can do this in a little more detail. It's probably worth going over an example where somebody has done this in a little bit more detail, because I think the questions I'm asking about describing what happens or trying to understand why things are happening, those are, I think it's challenging if you haven't tried those kind of exercises before to know what's expected of you in terms of an answer. And again, it's a little bit like asking how long should a paper be, and the answer being long enough. It's not that there's a perfect answer for that. There are legitimate differences. But there are things to think about in terms of analyzing a problem of this kind. And I hope that as we go forward through the semester, people develop some sense of what a reasonable answer is. Not necessarily, I mean, there can be a wrong answer. It could grow and you say it shrinks. It could, your code could crash. Those are non-answers. But more, if you see a particular pattern of behavior in the number of cells, the pressure, the pattern, the size of the cells, those patterns, what is a reasonable thing to say about those patterns? And to some extent, those are things that you have to develop an intuition for. And if you're an experimental biologist, you do a lot of microscopy, you look at a lot of pictures of cells. and you develop some sense of intuition. If you're a pathologist, you develop a sense of intuition about what is it that makes the cell cancerous and non-cancerous. You score it. If it's dynamic and you're looking at the living tissue, you have questions about how are the cells moving, what are they doing? And it takes time to develop a vocabulary for describing those things. And people do that in different ways. People respond to these images and ideas in different ways. And I hope that with time we can do that together and help people build that kind of understanding. So there were some questions about the other homework problem. And the other homework problem was we started out with the volume of the cell growing at a constant rate. And then I suggested that you have the volume of the cell grow at a rate that decreases as the pressure increases. And one thing you have to be a little bit careful about is that the sign of the pressure may be opposite to what you expect. And so maybe P has to be minus the value that you get from Compusal's pressure. And so that's something you have to look at. And I suggested that there were a couple of possibilities one could do. And this was something we started with at the very end of class but didn't do. And it was look at the growth rate and say that instead of growing at a constant rate, it was a constant times a decreasing function of the pressure. And the pressure in this case is whatever the number is that increases as the system gets crowded. And so if that's a negative number, you have to put a minus sign in front of it. And I suggested doing a linear decrease. Josh, go ahead.
Yeah, I was kind of confused just with the notation just in the problem. So you never really defined, so like in part A, define a linearly decreasing growth rate. What is V, the volume? And also, what is the reference pressure? I didn't totally understand just from that what those values were.
Right. Okay. So those are good, excellent questions. So here I've drawn schematically three possibilities. And in this case, I'm assuming that as the system gets more crowded, the pressure goes up. And the axes are actually not labeled because when I copied it, the axis labels got lost. So the X direction here is the pressure. So if the cells are under pressure, and the y-axis is the growth rate. And so in the first case, if the pressure on the cell is 0, whatever that means, the cell grows at a rate of 4.5 or 5, whatever it is. When the pressure gets to 5 here on the x-axis, the growth rate is 0. And in principle, if the pressure gets higher, the shell actually shrinks rather than grows, although that you might want to truncate that. Now, I said in the homework problem, I believe, that you needed to do an evaluation and see what was a reasonable value for Pref. And what will happen in this case is if you make Pref too small, I'm shifting the point at which I go to 0 to smaller pressures. And that means that the system won't grow, or it could shrink and all the cells could disappear. If I make Pref too big, then the system will grow, fill all of the space, and it would be as if you didn't do anything. And so my suggestion was, here we can calibrate G prime by doing a simulation with many cells but with no growth. So you basically create a cluster of cells with no growth, and you say, what's the pressure in the cells? And that pressure is going to be something that the cells are, in quote, comfortable with, would be normal. And so you would say that that would be a good place to start. If in that situation, the cells should behave as if they're not under pressure. And that would be something you could use for PREF. You might make it a little bit bigger, you might make it twice as big or half as big, but that would be a place to start in your exploration. And it does that, and so that would be the way I would do it. And we can do that together now. That was why I wanted to do this in class together, to see how people were able to handle it and work through it with you. I wrote these functional forms in the way I did. They're actually not quite right because they're not consistent. After having said that, I was going to make them consistent. They're not. The first one really should have been K times 1 minus P over PREF to be consistent. The other two are designed so that if P is 0, you get a value of K. And so the first one should have had P rescaled by PREF instead of the difference to be consistent. We have this semester talked about Michaelis functions and Hill functions. But one of the things that you do a lot in computational biology is you want situations in which values are bounded and either go up to a maximum value or down to 0 in a controlled way. And the functional forms you typically use look like the one in the middle here, PREF over PREF plus P. If P is 0, I have PREF over PREF, which is 1. If P goes to infinity, I have PREF over P going to infinity, which is 0. So this function, PREF over PREF plus P, for a positive value of P, is bounded at 1 at 0, when P is 0, and decreases to 0 as P goes to infinity. It's a monotonically decreasing function and has maximum slope at 0. And that happens to be called a Michaelis function because the chemists use it in a different context. It also describes the rate of certain kinds of chemical reactions. If I want things to be, if I want my slope to be 0 at 0 and move the place where I have maximum gradient to another value, I use what's called a Hill function. And the Hill function looks essentially identical to what I had here, but it raises each one of these terms to a power. Notice that if I set n here to 1, I come back to the one on the left. If n is a number like 4, what I get is something that begins to look, is called a sigmoid. As n gets bigger, it becomes more and more looking like a step function. If n goes to infinity, it's 1 up to PREF, and it's 0 for P bigger than PREF. And so this is a smooth interpolation between a step function and a linear function. And this kind of functional form is used a lot in situations where we don't know the details. Both of these have the property that if P equals PREF, I have PREF over 2 PREF, I have 1/2. So always at P equals PREF, the value of my function is 1/2. And so that's why PREF is a reference pressure in this case. I don't know if that helped answer the question at all. And so my assignment was to start out by using this linear decrease. And when you do the linear decrease again, you have to decide what happens when you get past the point where it crosses 0. These two guys have the advantage that they never go below 0. You don't get oscillations. This one you don't. You have to decide what to do at 0. What you typically, I could tell you what the answer is a little bit here. You'll get a fundamental difference between this one and this one, but I don't want to give that one away. Joel, go ahead.
Yeah, I have such a weird results between the Hill function and the linear function. On the Hill function, I had something, I think it's now looking at it, I think I changed the sign for the absolute value. I put like minus instead of a plus that is in your slide. And what I had was like one cell would go on this spree of dividing and it would divide into, I don't know, 5 different cells. And the other ones look like frozen in time. So the shape that I have on the tissue is completely different than I ever seen. I don't know if I could keep that in the report just so you can take a look on it and try again with the correct plus sign instead of a minus sign. But it was an interesting exploration of how weird it can be.
Well, so there are a couple of possibilities of how to do this here. It depends a little bit on how people did with this assignment. My next slide would be basically saying, use what we wrote last time and implement this, which I can do. We could do that together. Or we could have Joel show us what he did and look at his code together and see what's going on. What do you think would be most useful to you? I fundamentally, my goal here is to try to help you get through this idea and understand it. And I'm quite open to your feelings about what the best way forward is. I don't have a slide deck. I don't have a set of slides to show you the answer, except to assign it to you. And so you tell me if people would like to work on it together. Did everybody try this problem? Is there anybody who didn't try the problem?
So I'm just still confused. So in part A, you say growth rate as a function of pressure is equal to V times Pref minus P. Is V just a constant and Pref is just some random value that we choose to set the slope, or?
Well, it won't set the slope, it sets the intercept. V sets the slope and Pref sets the intercept.
Okay, so yeah, I think that was just my biggest confusion because I wasn't sure. You just say change both V and Pref, but you never really say what V and Pref need to be. So Vref or V is just the slope, and Pref is just a reference pressure that we choose.
Right. So K here would be. I switched symbols on you. I had V instead of K. So that was the switch, but the structure is the same. And whether P should be whether the pressure term that you pull out in Compusal should be P or minus P is something you have to look at. But yes, your basic interpretation of the assignment was correct. And so we can do it together if you like. I have no problem with that. Or you could show your code, or Joel could show his code. Again, you tell me what's useful. Joel, do you have it running for you?
I don't have it running, but I have like the document. If you want me to show the document, I can also pull it up and run. Here, let me just, if I can just show you what I got. And it's weird because the pressure goes to 0, which is something that I was not expecting. And the growth, like everything starts well, but I think there is a problem of the sign. I should try the sign different. I don't want to make the class all about this problem here, so I think other people have other questions. Maybe we should do together, and maybe I can find my error somewhere.
Okay, well, again, did anybody else have issues with this problem?
I had similar confusions to start. I can't say that I got a chance to work through it all the way though, because I ended up prioritizing the presentation and only got to spend a subset of the time that I would have poking around with this problem, but I had similar confusions on the notation. So I did not get a chance to work through this one fully.
Okay. Let me see what I can do. Does everybody have the code we did last time for cell growth that we did last time in the class? Let me see. Let me see if it runs. I have it running, but I better before I tell you that I'm going to do it with you, I better make sure it works. So let me make sure it works before I do anything else on my end. Okay. So I guess everybody should try to fire up Compusal, whether it's on NanoHub or otherwise, and go back to the code you had from last week. I have the version that I wrote available here, but you would have your own. And let me screen share that, and we can look at it together. And I want people to don't worry so much about all the plots. Worry about the basic dynamics of the problem. So why doesn't everybody take a minute to get a simulation that looks a little bit like this? Whether you plot the surface tension, the volume, the pressure is not so critical. Whether you're plotting here's the pressure field. I'll turn the pressure field there. But something where the cells grow. The cells grow. You can monitor the pressure, and you can see what's happening. And the details of how that is, I think, are not. This is for okay. Joel, question?
Oh no, I was about to turn something on my phone, but I can tell you that I changed the sign for the hill function, and things are working fine now. So it was just a sign problem that Compusal didn't like and get a weird result.
So can people see the simulation now? So you'll notice that the pressure is returned by Compusal in the center of this tissue is minus 183. And so that means that the Compusal is returning a number that's negative when you're being pushed on. So that's already telling you that you probably want to use minus the pressure value rather than plus. And again, pressure is defined as a directional thing. And so it's not wrong to say the pressure is in that direction, but it may be not the most natural thing to do, because if you're being pushed on, you think you're under pressure. So this is a simple growth simulation. And let's come back to the rule here. The rule here was that the cells grew at a constant rate. And here I was focusing not on the cell growth, but rather on keeping the target surface well behaved. So it was an earlier problem than the one that we're dealing with here. And so to make this a little bit more interesting for ourselves, I'm going to, let's see, this is growing by 25/50, a half of a voxel per monochrome step. That's pretty fast. So maybe that's enough. And probably in order to make things a little bit simpler, I'm going to turn off the surface tension entirely in the system. I'll leave it in there, but I'm going to turn it off. So we're doing a simpler problem. Let's try running that. Okay, I see my cells growing. They're dividing. I can zoom in here to see what's going on. They're growing. They reach a doubling volume and they divide. And I could visualize the pressure field. And I notice that the pressure is larger in magnitude in the middle than the surface. And you could think of this, for example, if you're making meringue, or you have honey, something that's viscous, and you pour material into the center, it piles up. If you wait, it'll spread out. And so if I had the cells growing more slowly, I would see that the pressure would be more uniform in this tissue. And part of the reason this is going so slow is because I'm plotting the volume at each time point for each cell. And the number of points in these plots gets very large as it runs. And the replotting takes time. This is what Alex is supposed to be helping to fix. So this is an issue with the plotting function. But we can live with that for the moment. So let's start with that. And let's look at what we have here. And again, I'd like people to be trying to do this on your own too. Let's look at the division rule. The first thing we see here in the mitosis steppable, if cell volume is greater than 50, we have the cell divide. And let me try. One of the assignments was saying, instead of saying cell volume greater than 50, we said cell target volume is greater than 50. Right? That was one of the homework assignments. So let's try that, see what happens. Let's come back to see what we had before that. What I see before that is that the cells are growing in sync and dividing and growing in sync and dividing and growing in sync and dividing. And then as time goes on, I see scatter. And the reason for that is that the cells in the middle are not growing as fast. Even though their target volume is going up at the same rate, they're not growing as fast because they're under pressure. And so they're dividing more slowly in the middle than they would on the surface. And if I look at my pressures, I should probably put a minus sign in, but the cells at the boundary are these ones with low pressures, and the cells in the middle have bigger negative pressure. And there's something else to notice. Where's 0? Notice 0 is above this. Almost all the cells have negative pressure, even the ones that are on the surface. And the reason for that is that there's a positive contact energy between the cells and the medium. And that means that the medium is always pushing inward on the cells a little bit. And the reason I mentioned doing a calibration by simulating with no growth was that 0 is not really the right reference. There's some reference value that's not quite 0 that you have to know about. And so in this case, if I look at this picture, I come across this line, 0 is above all those points. If I wanted to know what's the typical value when there's no growth, it's somewhere between 0 and minus 100. It would be minus 50, maybe. And so that would suggest I would use a P-ref of minus 50, minus 100, minus 25. So that's how I would think about that problem. And we'll do that in a minute. Let's first come back and do the case where we have the cell division. Let me let this run out and let's run into the, it may not, because of the replot, it may wind up being too slow. The reason I originally said to use a smaller lattice was because I didn't want people to have the simulation to take too long to run into the edges. Let's give it another minute or two. If you'll notice that the cells in the middle are a little smaller than the cells on the boundary, but all the cells are roughly the same size. They're not hugely different in size. And I'm a little frustrated that NanoHub is not giving me more CPU cycles at the moment, because this is running slower than it should. Okay, I don't think I'm going to wait for it to run into the boundary. You can do that at home and see what happens. So now let's try it with the one where we have cell division on the target volume. In this case, you'll notice that everybody divides at exactly the same time, because the target volume is independent of the actual volume of the cells. You'll notice there's a big scatter of actual volumes. See, everything divides synchronously. Notice that the pressures get pretty complicated after a division, and then they rapidly equilibrate. And I'd like to see, I maybe should change the simulation to put it in a smaller lattice so that it doesn't take quite so long to run. Because this one becomes most interesting. Let me do that. This one becomes most interesting when you have the cells bumping up against the boundaries. I'm going to change that. I'm going to make the lattice smaller. 100 by 100. I have to recenter my initial cell. Everything else is okay. So let me try again. Let's watch that together. The cells are growing, dividing synchronously. And synchronized cell division happens in biology, especially in early embryonic development. You get a lot of synchronized cell division. In adults, not so common. Okay, I'm now going to start running out of room. Let me switch to the pressure field so we can see what's going on. Notice in this case, the pressure is not uniformly higher in the middle. It's distributed. And as things keep going, the pressure is going to keep going up and up. And the size of the cells will get smaller and smaller. Let's give it another minute to fill in those corners. Minus 282 for the maximum pressure. $174. Certainly doesn't look very biological. It's a little hard to see, but you're actually having cells that are disappearing. Some of the cells are getting squeezed out of existence here. If I zoom in, look at these little blue guys here. And the steps are getting pretty slow. If I were doing this in homework, I'd ask you to plot the number of cells, maybe count how many cells disappear per time step. And if I look at everybody just divided again, notice the average volume of the cells is going down as time goes on because the number of cells is going up. If I go back and switch back now that I'm in the smaller one, let me come back to that other rule that I had where I have the cells divide on volume again rather than target volume. And this time with the smaller one, I can run it all the way through. So let me restart. And I'm doing this partly because I want to talk you through a little bit some of the things you might look at when you're asked to describe what's happening. But at the beginning, it doesn't look very different. Let me put this on pressure so we can see the pressure field. Notice the pressure behavior is totally different for this from the other one. Here, the pressure is concentrated in the center of the aggregate. The range of pressures is different. Let's see what happens to the volume when I fill all the space. Is it going to keep going down the way it did before? And what happens to the maximum pressure? And the surface tension plot isn't doing anything because I turned off the surface tension. You'll notice it takes more Monte Carlo steps to fill the lattice here than it did in the other one because the cells aren't all dividing at the same time. Cells on the surface are dividing faster. And I apologize for keeping you waiting to watch NanoHub do the computation. It'll only be another minute or two. Is this making sense for people? Because if it's not useful, tell me, and we can move on to something else. I don't want to waste your time. Okay? The experimentalists who spend a lot of time looking at cell culture or tissues growing in vivo will be used to watching cells do their thing. As an experimentalist myself, I love watching experimental data. I can watch movies of tissue development again and again and again and again, because you learn something every time you see them. And I think the way our brain processes movies is different from the way it processes still images. And to me, one of the most valuable things in something like Compusal is the fact that you can watch the dynamics. And I should probably, Giuliano, you should probably be helping me show them how to do a movie, because saving these data sets is something. I've been asking people to do screenshots, but Compusal will let you, if you go under settings here, and go under here under output, if I click save image every n Monte Carlo steps, it will save the output to a file. And unfortunately, it'll give you a stack of still images. And then depending on whether you're on a PC or a Mac, you have to use, on a PC, it's very easy. You select all of them and say convert to movie, and it'll do it. On a Mac, I think you have to use, there's a program you have to use to do that. The other thing that you have to do when I say save image every n Monte Carlo steps, which is a little bit of a cheat, is that you have to hit this camera button in the window you want to save, or it won't save it.
That makes so much. I literally was trying to track files for like an hour to make movies happen the other day. So that is very good to know. That was the missing link, I think.
I find that confusing, and it didn't work that way in the old system. But the issue is you don't know which. In principle, you could have many, many windows. And it might be better that if in the settings you selected which window you wanted to save from, but instead the idea was you went into the console, you hit the ones you wanted to save. And the idea was that you might, for example, have different visualizations. You might have different color settings. And you don't know which one of those you want, so you click the ones you like to save. So that's why I wanted to do that with you, because I realized that we hadn't covered that. And it's a basic thing. Giuliano, we should make a note to put that into our manuals a little more clearly. Maybe put in the class a little earlier too. So now I'm seeing right in the middle, I have a high pressure region. Boundary is still low pressure. I'm beginning to fill my space. Notice that while I'm getting some small volume cells, the average volume is not going down the way it did when I divided on target volume. My average is still around 30, maybe going down a little bit, but not a lot. Notice the pressure now, however, is going more negative. There's still a little bit of red here, which is unfilled space. And if this were just time step 2 or 3 more time steps, we'd see everything there is to see, but it's just slowing down on us. 570. Okay. So now I filled all of the space. And again, I will see that some cells actually are getting squeezed out of existence. Their volume is going to 0. And so there can be some instabilities where you get patches of cells that collapse. Biologically, that happens in some situations, but it's not a normal biological process. It means our pressures are way out of line.
So between these two cases, when we're using target volume as opposed to actual volume, are the ones with target volume more prone to collapsing because they're continuing to divide? Okay.
Yeah.
That makes sense.
Because you're pushing them closer to 0. And when there's a separate rule. I mean, in principle, I suppose you could keep a zero volume cell, but it's not clear what that means. Compusal automatically sweeps any cells that have zero volume and says they disappeared, takes them out of the world. And so in this case, it's actually rather something that you could explore. If the growth rate were slower, what you would find is that eventually all the cells will have the same volume and everything will stop. If the growth rate is too fast and the lambda volume is not big enough, you'll get always some cells disappearing somewhere, and so there's always going to be some growth somewhere and some disappearance somewhere. And you'll get waves, pressure waves moving through the system. I think I can't tell you whether this particular pattern that I've picked is going to equilibrate or not. It looks like it's getting more uniform. If I zoom in, I see that there's some big cells in the middle, but most of the cells are almost the same size. All these cells around the edge are almost the same size. And so this system here is going to, I think, eventually come to a situation where all the cells are about the same size. Nobody's going to divide anymore. Pressure will keep going up. So it's going to reach a pattern that's constant, even though the pressure will keep going more negative. And so those are really dramatically different outcomes. Oops, sorry, I don't mean to be zooming the wrong way here. And you'll see that here in the volumes. Here, between time 550 and about 600, there were some cells that had very small or zero volume. Notice now all the cells are within a range of 10 to 30. And so when I'm asking you what to describe, these are the kinds of things you might describe about what you're seeing the system do. Now you could ask the question then, why are they doing that? And we could explore that together. They're doing it in this case because all of the cells are under pressure. The system is becoming mechanically equilibrated, and so the pressure is balancing out. Right? Notice the range of pressures is going down. The dispersal of the pressure is going down. The dispersion of the pressure is going down. Dispersion of the volume is going down as well. And so this system is actually becoming rather plant-like. The cells are under very high hydrostatic pressure. They're not going to move. Maybe a little bit, but not much. And so the system will freeze up. It'll lock up. So again, there's a dramatic difference between the two results. Any questions about that? Suggestions, ideas? Okay, let's look back at that. Let's try that other exercise. Let's go back to the editor. And I better hit pause here. And let's think about what we can do with the growth rate. So our growth rate was here. We're growing by half a voxel per Monte Carlo step. While I'm at it, I'm going to flip the sign of the pressure. I'm going to put a minus sign when I display the pressure so that it's positive. And so let's look back here at what the pressure was under what we call normal circumstances. The pressure was about minus 50. So let's use that for PREF, 50. I'm going to flip the sign. 50, reading it off of here. And I'm going to say that my volume was my 1/2 voxel per Monte Carlo step times 50 minus the pressure, but that's negative. So it's going to be minus minus is plus cell dot pressure. And I do have to think a little bit about whether that's this first one, the linear one. And I do have to think a little bit about what I want to do if the sign of this is the wrong way. Do I want cells to shrink if they're under the in the wrong direction or not? Well, let's try that. That's the exercise I started with. Let's see what happens. And let me also on my plots, I'm going to only plot every 10 Monte Carlo steps. So that it doesn't, the plots don't take so much time. Because it's the plots are what's slowing down the simulation is plotting that data. Any questions about that? Does that make sense now? So far? So I'm trying to do this first exercise. And who knows, I make all those changes, it may crash. I would not be the, I'm quite capable of writing buggy code, I promise. Yeah, I have a problem that my cells are not growing because of what I did with the pressure. So that didn't work. So let us try, I'm not going to let them shrink. So I'm going to do max of this and 0. And I probably still should have it. I want them to grow. I think. I'm not sure what this will do, actually. I have to try it. And the fact that the window resizes every time you restart is a bug. Earlier versions of Compusal didn't do that. And okay, now that's better. So this is what I wanted. Okay. So now I'm getting rapid growth. The pressure is going up. And notice the pressure is saturating. It's not going to infinity, it's bounded. So the system is equilibrating. I don't know if I'm getting any cell division here or not, but everybody's coming more or less to equilibrium. So that's a reasonably well-behaved result. What happens if I change that threshold? I made this 50. What if I make it 25? Is that going to speed up the growth or slow it down?
Let's slow it down, right?
Slow it down. If I did it right, maybe I did it wrong. It's still growing fast, but not as fast. And notice the pressure is staying bounded. It's not blowing up anymore. Everybody's getting to be more or less uniform size. What happens if I now trigger on the target volume rather than the actual volume with this rule on pressure? What's going to happen? Somebody guess? Somebody have it working? Again, part of the reason that I give you these open-ended exercises, and I understand writing the code initially and getting it working takes time. But one of the things that's nice about Compusal is you can explore opportunities. The cost of trying again with a different parameter value is not very big. And so I understand that writing the code to do the display and takes time. But once you have it, trying different parameter values is not expensive for you. And so one of the reasons I ask these open-ended questions is to try to encourage you to play with this. It may not be the most exciting game in the world, but you can play. And so let's try just doing the target volume here. Let's see what happens. It should probably have been displaying the pressure. You'll notice that this time, even when I trigger off the target volume, the difference is not so big. Because if the pressure gets to be large, my growth of target volume stops. And so the system is still more unstable. You'll notice there are always some cells dividing. These guys are getting big, other ones are small. But it's not that dramatic difference between the two results that I had before. See, the variance of the cell volumes is very large here. Pressure range is a little bigger, but not that big. It's still staying bounded. And there's really a lot one could do. I mean, one could, in principle, there's a lot to explore with these systems. Whether those systems are in fact telling you something biologically meaningful is another question. It'll depend a little bit on the biology you're interested in. But it does give you a sense of what the sort of the biophysics of the problem can be. And one question that definitely is of interest is when you run these things for a long time, what happens? Does cell division stop? Do cells always divide? Does the volume go to a fixed value? Does the width of the distribution go up or down? Does the maximum minimum pressure change? Does the mean pressure change? Those are all questions that you could ask when you look at these systems. And I could make a long list of those in the homework problems, but my experience is that making a long list, everyone ignores. You've had a few homework problems where I've had long lists of things to consider. And so I'll do that a few times, but I think it's important to try to begin to develop a sense of what you think is important. But until you look at the time series and see what's happening, it's hard to know what's important. But stationarity is a good question. Widths of distributions are good questions to ask. Is the system dynamic or static? Good question. So now I'll leave it to you, I think, to do these other two cases. But I hope that that gives you a place to start in thinking about the problem. The one thing that I might do if I were going to go between these cases, let me just show you one thing that I would do to make things a little bit easier on yourself. Here I said this is Pref minus P. I would rewrite this as 25 times 1 plus cell pressure divided by 25. I haven't changed anything, right? I've multiplied and divided by 25. And let me double check to make sure I didn't mess it up. And again, I realize that I try generally not to code in front of people because I don't think it's usually very informative, but I hope that once you'll excuse me for doing that, to walk you through the way I think about the problem. That has a very fancy educationalist name, which is cognitive apprenticeship. I'm generally not a big believer in educational buzzwords, but it basically means doing things in front of you and hoping that something rubs off. Okay, so I didn't wreck it. It's doing the same thing. So now I could go back, and now that I have this form, I could rewrite it in this other way. I could rewrite this as, instead of 1 minus that, I can do it here. I have, sorry about this, give me a second to write this correctly. I want to write this now Pref over Pref plus P. So I will write this as Pref, which I made to be 25, divided by, parenthesis, 25 plus, except plus is a minus because it's a negative sign, minus cell pressure. And I have to be a little bit careful here because this could, because cell pressure could be, I'm going to have to do a max of this and some number, because this could go to 0. I don't want to have it divided by 0. Sorry about that. Otherwise, I could take the let me do the absolute value. It's cleaner. And then it's a plus. I don't want 25 minus pressure to be 0. I don't like pressure. Yep, I didn't. Probably a parenthesis, huh? Line 56. What did I do wrong? Giuliano? It's gotta be a parenthesis. 1, 2, 3.
You're missing one closing parenthesis, I think. You have four opens and 2 closes.
I don't need a max here, for one thing. So 25 over 50 times 25 times, I don't need this 25. 25 divided by open 1, 2, 3, 2, 1, 0. No close parentheses. Is that right now? Okay, this is this middle one. Doesn't look that different, does it? Pressure now is not saturating them. Those pressure's going down, not saturating. So that one's not shutting off as well as the other one did. And the last one that I'll do is if I want to do the exponentiation, I'll do fourth power. Okay. So I don't want to take more of your time. I've used more time here than I had planned on this, but I hope it was at least a little informative. Let's take a 5-minute break. And when we come back, we will start talking about chemical fields and diffusion. Okay? Any questions before we break? Suggestions? If people are having trouble replicating what I did, come back to me. But I thought walking you through how I would do that homework problem, I think Josh asked very good questions because the problems were sort of intentionally vague, and I understand the how that can be confusing. But I hope this is also a way of thinking about showing you how to think about a problem and being experimental, running the simulation, seeing what happens, trying out a different value, trying it again, and trying to build the sense of what's reasonable. And I understand to say figure out what's reasonable is really a threatening thing to do in a homework assignment. If you're used to problems with definite answers, the answer to ask you to say what's reasonable is I'm sure you've had problems like that, but it's not typical in an introductory physics course. It'll say find the value of x. It won't say what's a reasonable value of x. On the other hand, when you're doing research or engineering, what's a reasonable value of x is usually more of the thing you have to do because you don't know the answer in advance. And so developing a sense of what's a reasonable answer, I think, is a critical part of this course. Okay, all right. So now let's talk about concentration fields and diffusion. Now, for some of you, this will be a review. You can help out everybody for whom it's not a review. And there's probably a little bit too much detail here, and so we may skip some of the detail. You can go back to the slide deck. So the first thing to think about is what concentration means. And we're going to use the concept of concentration pretty loosely in Compusal. And on the length scales that we're working on, typically it's a reasonable concept. But biologically, concentration is a little bit problematic. When we're dealing with molecules like water, I mean, of course, ultimately, atoms and molecules are objects. They're discrete objects. Concentration is treating discrete objects like continua, and that means that there has to be a length scale over which you can draw a box where there are a lot of molecules of a given type in the box, but that's small enough that the concentration doesn't change dramatically from within the box. And so concentration represents what's called a mesoscopic length scale. And these pictures come from that textbook that I recommended, The Physical Biology of the Cell, that beautiful book by Phillips, really beautiful books by Phillips and Therio. Julie Therio is one of the great experimental biologists in cell biology. Did very pioneering work imaging actin cytoskeletal dynamics. And Phillips is a great theoretical physicist at Caltech. And having that double viewpoint of the experimentalist and the modeler and the theoretician, I think makes that book better than any of its competitors. But in any case, we have to think about this concentration field. Now, typically we will use this idea of a field loosely, but biologically you'll have many molecular species which exist in very low copy numbers. Take DNA. You have X chromosomes or X molecules of DNA. To talk about the concentration of DNA in a cell is a little bit problematic because you don't have enough molecules that you can average. And there are other species too, things like transcription factors, which exist in very low copy numbers. And so talking about the concentration of a transcription factor, which we will do, may not be so sensible. If you're talking about calcium, there are plenty of calcium ions, and therefore concentration is reasonable. Water, reasonable. ATP, reasonable. But for many enzymes and signaling molecules, concentration is a little bit problematic. And for macromolecular machines like actin filaments or microtubules, where there's spatial structure, the idea of concentration doesn't make any sense. And so physics lives in a world where things are homogeneous, where you could average. And in biology, very often averages don't make sense. And so we have to remember that physical concepts like concentration, viscosity, even pressure, only make sense in a world where you can average, and biology doesn't always give us that. That said, it's a useful concept and we'll use it. But at the back of your mind, you should be questioning, in particular biological situations, whether concentration actually makes sense. When we have fields, you could have other kinds of fields. You could have chemical concentration. You could have electrical potential. You could have forces. You could have cell densities. So there are many, many things that can be described as continua or fields. Fundamentally, a field is simply a number which has a different value at every position in space, a scalar field. In this particular case, for molecular densities, we'll define the number of particles within a box The box has a volume of V. The concentration would be C divided by delta V, by the volume. There are many ways that chemicals can be moved around in a cell. We are only really going to be able to address one of them, which is diffusion. And so I will just very quickly alert you to the fact that there are other kinds of transport in cells. One of them is advection. If you have a flow, then chemical concentration is carried by the flow. If the flow is non-uniform, and this is a classic example of a pipe flow, so there's a wall here, so that there's no slip boundary conditions. The flow velocity is zero at the boundary. In this case, an initial radial distribution of concentration gets stretched to make this sickle cell or arch-shaped distribution. And those kind of what's called advective transport is something that's very important when you're dealing with things like blood flow or lymph flow. And it happens also within cells under certain circumstances. You could have active transport of cytoplasm as well. One of the things that we're going to face in Compusal is that the chemical fields are going to live in their own lattice. They're not going to live in the cell lattice. And so when cells move, they don't automatically carry chemical concentration with them. If cells move slowly, that's not a problem. But in the region where cells would be carrying material a lot, this could be a problem. Compusal in principle allows you to do chemical fields where the cells carry the chemical field, but that's not as well developed in Compusal as it could be. And so typically within this course, we're going to assume that we don't need to do that, but it's something to remember. The other thing that you have in biology is active transport. And active transport comes in a variety of forms. At membranes, both between the cell and the outside world, and between the inside of the cell and the mitochondria, the organelles that create energy in the cell, and potentially also between between compartments in the cell of other kinds. You have various kinds of ion channels that cross the boundaries. Some of these ion channels are basically holes. You say, why build a wall then drill a hole through it? Well, we could talk about that. But some of these ion channels actually are pumps or selective. And they can do very complicated things. They can act as water pumps. They can do what's called antiport, where one particular molecule, in this case calcium, moves out, and whenever one calcium goes out, a sodium comes in. You can do symport. Here, one molecule of sodium comes in, and that brings one molecule of glucose in. And so there are a variety of kinds of pumps. And those pumps can depend on things like ATP, the energy currency of the cell. They can depend on voltage. They can depend on chemical concentrations. And one thing that we will face in Compusal is because the membranes themselves don't have an identity in Compusal, you can't attach the pumps easily to membranes. And so our ability to do active transport across membranes is limited in Compusal. It's not zero, but it takes some work to make that happen. Another kind of active transport that you have within cells, so that's cell transport between cells in the outside world and vice versa. Another kind of transport that you have within cells is what's called vesicular transport. And recent Nobel Prizes have been issued on some of this. And if you'd asked me 20 years ago what one of the big mysteries in cells was exactly this. Cells have we haven't really talked about the intracellular structure of cells. I had a lecture on that, which we've skipped. Have a variety of filamentary molecules inside of them, macromolecules. You've heard me use the terms before without really defining them. The main ones that you run into are actin and microtubules. The third category, which people tend to ignore, but is also very important, are what are called intermediate filaments. There are molecular motors that move on these filaments. And there are molecular motors that move, the filaments are polarized, they have a direction. And there are molecular motors that selectively move towards the plus end or the minus end of these filaments. Those molecular motors in turn can anchor lipid vesicles, so volumes of fluid that are surrounded by membrane. And that means that they can actively transport material like little railroad cars running down a railroad. That winds up being extremely important when cells get large. In particular, we wouldn't have neurons if we didn't have this. Because what we'll find is that transport by diffusion slows down as things get bigger. It slows down as the square of the size. And so diffusive transport works wonderfully over scales of microns, but doesn't work effectively at all over scales of centimeters. And if you have a neuron which has an axon length of a meter, and you ask how long would it take for chemicals that are synthesized in the main cell to get to the end of the neuron, the answer is tens or hundreds of years. With active transport, the time scales linearly in the distance, and so those things become possible. Again, we are generally not going to model active transport, but we need to be aware of it, and when it's there, we have to think about how to deal with it. Another thing that happens, which is related to cells taking things in and getting and eliminating them or releasing them, and has become very important in people's minds recently because it's one of the ways that COVID gets into cells and out of cells. Many viruses do this, is what's called endocytosis or exocytosis. So one of the ways that cells take material from outside the world is through direct diffusion across the membrane. Small molecules like water move that way. Through ion channels and pumps, holes in the membrane of various kinds. Another thing that they could do is that the membrane can actually pinch off, surround a volume of material from outside the cell, and pull it inside of the cell. That's called endocytosis. And that's how SARS-CoV-2 gets into cells. Not all viruses get into cells that way, but most do. So they actually use the active normal processes by which cells bring things inside of them in a pathological way. Similarly, cells can take vesicles, membrane-bound regions of material. They can fuse that with the membrane and release their contents. And SARS-CoV-2 does that to release itself from the cell when it's synthesized. But that also is something that happens normally, for example, at synapses in neurons. Chemical signals at neuronal junctions are released that way. And so endocytosis and exocytosis are additional ways that cells move material outside the cell into it and vice versa. Again, in our world, this is a level of granularity we're typically not going to represent, but we have to begin to think about how we would represent those processes computationally. The main thing that we're going to focus on for the next class or two is going to be diffusion. And diffusion is a passive process. The things we've been talking about so far are all active ones. It's a passive process, and it's a result of essentially random movement and entropy. If I have a high chemical concentration in a fluid, and the fluid's not moving actively, and I let it sit, I will find that with time, the concentration gradient will decay, that the chemical that started at one end will spread until, if I wait a very long time, the chemical concentration is uniform. And the reason for that is fundamentally pretty simple. If the molecules here are moving randomly, and the molecules here are moving randomly, in this region here, the number of molecules moving to the left is pretty small. In this region here, the number of molecules moving to the right is pretty big. And so the net flux will be from high concentration to low concentration until the concentration is uniform. And we're used to this in the context of temperature, where heat flows from high temperatures to low temperatures to equilibrate. We're used to it in the flow of water, to flatten out the height of water. And we certainly are used to it in terms of chemicals. The classic example is you put an ink drop into water and the ink will spread. You'll also notice that if you do that, if you watch a video of that, it takes a long time for that ink to spread out over a reasonable distance. It's not fast. It spreads over short distances very quickly, but not over long distances. Diffusion is a little bit different in free fluid, like water, across the membrane, where there's a boundary, and within a crowded space like a cell. Cells are not full of water. They have a lot of water in them, but a lot of their volume is proteins. And it's more like a person trying to weave their way through a crowded subway car. You can't move freely. You have to go around things, and that changes things a bit. There's a beautiful old computer animation of diffusion happening. This was done by Harvard. It should have a label on the movie. I need to add the source for that. It's quite an old movie. But this is showing some of the complexity of random movement. And what they've shown here is a cell membrane. The membrane is oscillating, and you see various molecular species, and depending on their size, the repeat here is a little bit jarring because it's playing a little bit too fast. But different species move at different rates. Bigger objects move more slowly. Things that are attached to membranes move in a different way from things that are free. But there is this concept of random movement and distribution that's happening at all length scales. Again, a key thing to remember about diffusion is, one, that bigger objects diffuse slower. Two, that if the viscosity of the medium is higher, things move slower. Things move faster in water than they do in molasses. And both of those are given by if you want to get a rough estimate, by the Stokes-Einstein. It's a beautiful visualization, and I apologize for not having the reference. Maybe Giuliano, you could try to dig it up. It was done at Harvard. It's done about 15 years ago. It was really beautiful computer simulations that they did. And I know the earlier version of the slide had the credit for it, and I apologize for not having it. So larger objects move more slowly. Higher viscosities move more slowly. And in diffusion, again, the distances, it slows down with distance. So if a molecule takes 1 second to travel a micron, it takes 4 seconds to travel 2 microns, and it takes 100 seconds to travel 10 microns. And so that quadratic slowing down is a critical limiting factor for diffusive behavior. It used to be thought that bacteria only had diffusive transport. In fact, they do have some motors in them. And because of that, they were limited in size. In fact, there's some bacteria that get quite large, and they do have active transport in some situations. But in eukaryotic cells, this kind of active transported material within the cell and between cells and the outside is critical for the ability of cells to organize. And we certainly wouldn't have multicellular life if it weren't for active transport. You couldn't have large objects. Algae in the ocean, seaweeds, typically can be very large in two directions, but they're very thin. And the reason they're very thin is that they need to be all of their mass has to be close to the seawater because they exchange with the seawater by diffusion. And so you can make thin sheets that are very large, but they can't be cubic. You can't have spheres, big spheres, without active transport. And boy, you could to really delve into these questions would be worth take a couple of lectures. And at the end of this slide deck, there are a bunch of old slides from other courses that I've taught that go into more detail about the mathematics and the biology of these problems. Okay, so let's define the concept of a concentration, assuming that it's meaningful. We're going to count within a volume how many particles we have, n, within a volume. And we'll say the volume is centered at x, y, and z. It's going to be a function of time. And we're going to say that it's in a box of side delta x, delta y, delta z. And so the concentration is the number of particles divided by the volume. We'd like that to be well behaved. That is, it should change smoothly as we change the position. Clearly, if we make our box too big, we either have one particle in it or none. And so we're going to get the kind of thing we saw earlier that Connor and Drew were showing us, where the concentration fluctuates rapidly and randomly between locations. Concentration really only makes sense if it fluctuates smoothly. And so we have to have our box be big enough that n is big enough to make sense, that the fluctuations aren't too big. Similarly, we want our box to be small enough that we have a lot of boxes within our region. Otherwise, we're not getting any information. Okay. The flow of material per unit time at a given position is, for random movement, is given by something called Fick's law, which says that the flux is proportional to the gradient of the concentration. That is essentially equivalent to saying water flows downhill. And if you're not used to vector calculus, I try to avoid vector calculus, but this is one of the times when you'll have to put up with it for the next half hour. The only thing we really are going to do is vector calculus for in this class is to write the diffusion equation, which we'll do in about 5 seconds. And so this is saying that if you have a high concentration, material flows from the high concentration to the low concentration, and the rate of flow is proportional to the gradient, to the slope. And this introduces a constant, d, which is called the diffusion constant, which tells you how fast that flow happens. And the speed of that flow, as I mentioned, depends on the viscosity of the fluid, the size of the chemical, potentially on the electric charge of the chemical, the electric charge of the environment, the pH, temperature, many things. The units of this flux prefactor are distance squared divided by time. And that's where this concept of diffusion going like the square of the distance is going to come from. Diffusion time going as the square of the distance is going to come from. And so if the flux in and out of each boundary is d grad c, and I add up over all the surfaces, then the change of concentration inside of my box per unit time is the divergence of the flux. So del dot minus d grad c. If the diffusion constant is the same everywhere, the d comes out. Del dot grad is written del squared, the diffusion operator, Laplacian, c. Notice that the first equation is a vector quantity, the gradient of c, the slope of c, giving me a flux, which is a vector. Divergence is an operator that takes a vector and gives me a scalar. And so the diffusion equation says I have a scalar quantity, concentration, and I get the concentration change per time is a scalar quantity depending on the concentration. Del squared is the second derivative of the concentration with respect to x plus the second derivative of concentration of y and z. And this is the diffusion equation, and we'll use it repeatedly. If you have worked with the diffusion equation before, great. If not, the thing to remember is it tends to equilibrate concentrations in time. And so that's key. We are typically, and in Compusal, we have the ability to write a situation where the diffusion constant is not constant, where it varies in space. However, if the diffusion constant varies in space, we should actually be using this equation up here, where the derivative is taken on the diffusion constant times the gradient. And Compusal will actually use the second equation, where the diffusion constant changes in space but doesn't have a differential on it. That will give us potentially a small error in our numerical calculations. Usually it's not critical, but it's something to be aware of. The other thing to remember is that in biology, the diffusion constant's not only non-constant, and going back to that movie, that animation that we looked at before, is something to think about, but it's also not isotropic. If I'm in water, the speed at which my molecules go up, down, left, and right is the same. If I'm on a membrane, I can move along the membrane rapidly, but across the membrane slowly. That's anisotropy. Many biological materials, in particular extracellular matrix, have fibers in them, and those fibers are not always randomly oriented. They can be combed, where there's a preferred orientation. And in those situations, molecules move along the fibers much faster than they move perpendicular to the fibers. And so that means that D here actually is not a scalar quantity, but is actually a tensor. And so then you would have a tensor times a vector. And we're going to typically ignore that complexity. But it's something that, again, in some situations is important. And I'd rather tell you about the things we're going to ignore. And if you don't need to worry about them, you don't worry about them. But I'd rather let you know that there is something there to think about in the future. Another thing that happens, which is not put into this equation, is that many biomolecules bind to each other, not chemically reacting, but sticking. And in particular, a lot of signaling molecules, like growth factors, have something called a heparin binding domain. And that heparin binding domain sticks explicitly to collagen, which is in the extracellular matrix. And so that means that even though the molecule is pretty small and it moves around freely within the cell, when it's secreted into the outside world, it sticks and stops diffusing or diffuses very slowly. And so there are a lot of caveats to the idea that molecules diffuse in a simple way. With all of those caveats, it's actually rather amazing that assumptions of simple diffusion actually give you pretty good results in a lot of situations. But I don't want to undersell the complexity of the real biological situation. And I'm happy to come back to some of these details later. Again, Compusal allows you to do some of these things. Typically, people don't use those features very much. It'll make do with a simpler. Another thing, as I mentioned, is that the biological world is crowded. And so suppose that I have here a chemical that's sourced here and is diffusing freely. In a crowded world, the chemical can only go around the crowding objects. Here in this second picture, it's not only going around the crowded objects, but it's sticking to those objects. Clearly, the time distribution of that chemical will be different in these three cases. There's the added complexity that in the biological system, those crowded objects may also be moving around themselves. And there's some additional things that can happen, because if the things that are moving are not point particles but have finite size, there are phenomena called the depletion forces that have to do with whether these point particles are attracted to or repelled by surfaces. Again, we're going to neglect all of that complexity for the moment. So coming back to Compusal and fields. Compusal is going to represent fields as a lattice. Everything in Compusal is a lattice. Even off-lattice methods like Paul Macklin's Physicell represent chemical fields on a lattice. It's possible to represent chemical fields off lattice using particle-based methods, and they have some advantages and some complexities. So the software that TJ is working on, called Mechanica, does chemical fields off lattice with particles, and that has its own set of issues which we can talk about. But basically, just the way we take a picture of the cell lattice, and we say if we're in cell number 1, we put a number 1 there, and cell number 2, we put number 2 there. Here we take a picture of the chemical concentration, and we write in each box how much of the material we have, or the concentration we have. We could also have vector fields. Instead of having a number here, we could have 3 numbers, which represent an arrow pointing in some direction. And that's something that I think is underutilized. It's there in Compusal. It can be quite a powerful concept, and people haven't explored it very much. We are going to be doing diffusion, solving that diffusion equation. As I say, we're going to be solving a slightly wrong diffusion equation when the diffusion constant is not uniform, because we're going to be move it outside of the derivative. And there are going to be a bunch of numerical diffusion solvers. We're pretty much always going to use diffusion solver FE. And that solves the time-dependent diffusion equation. It's going to say, if the concentration is such and such now, what's the concentration at a short time later? And for most of the chemical species we're going to be dealing with, and the time steps we're going to deal with, that's reasonable. For some molecular species, particular oxygen, diffuses very, very fast compared to everything else. And for oxygen, in principle, we should be looking at the equilibrium concentration rather than the time-dependent one. And so there is a steady-state diffusion solver that runs in Compusal and gives you, if I ran the diffusion equation to eternity, what would my chemical distribution be? And the problem with that steady-state diffusion solver is it's slow. And so one tends to avoid it if one can. The other thing that can happen is that you have multiple chemicals in the world. They can interact with each other. They can react. Typically in Compusal, we'll do those chemical reactions in a different way, but it's allowed to write simple chemical reactions in the chemical fields themselves. And those are called reaction diffusion equations. And mathematical biologists love reaction diffusion equations. Geier and Meinhardt made an entire career writing beautiful reaction diffusion equations that did incredibly beautiful patterns and tried to explain all of biology that way. And if you look at the cover of a textbook like Murray's textbook, it'll show you pictures of leopards and then reaction diffusion equations that generate leopard spots. Whether reaction diffusion equations are actually responsible for biological patterning or not was sort of a vexed problem. There's some long history and interesting story about that. The person who invented the reaction diffusion equation in biology was Turing, Alan Turing. Of course, is famous for being one of the co-inventors of the computer. It was the last paper he wrote before he committed suicide. And it was called The Chemical Basis of Morphogenesis. It's one of the most cited papers of all time in biology. And he recognized that while diffusion by itself always spreads out variation, it always smooths things out. If you have two chemicals that diffuse and react together, you can get patterning. You can get a stable, an initially uniform system going to an unstable system of patterns. And that was a tremendous insight of his. And it's led, again, to a whole field of mathematical and computational biology. For many, many years, there was argument about whether it was appropriate in biology and when. And there was a very, very famous cover story in Nature, maybe 20 years ago now, 25 years ago now, which is one of these things that is so beautiful and so obvious, and yet has been under people's noses for thousands of years without being noticed, which was a study by a Japanese group of fish. And many fish have stripes on their bodies. And in the case of mammalian species like leopards, the spots on the leopard are fixed when the baby is very small. And then as the animal grows, the spot size gets bigger because the pattern is fixed and there's replication of cells. And you did a little bit of a simulation that was like this when I asked you to combine differential adhesion with cell division and type switching. That was to try to explore that particular pattern. And so in the case of a leopard, those big splotches were initially very small, fixed by diffusion lengths, and then there was cell division that led them to grow. In angelfish, as the animal gets bigger, the stripes move on the animal. The width of the stripes stays the same, and as the animal gets bigger, it gets more stripes. And that was sort of the definitive proof that Turing's mechanism really was there. And everybody who had these fish swimming around in their aquarium could have seen this, but nobody took a picture of the fish when it was small and a picture of the fish when it's big and said, gee, the stripes are the same size and they've moved on the body of the fish. It was one of those incredible discoveries that was totally obvious when somebody noticed it. And how many people had looked at those fish millions and millions and millions of people had looked at those fish. No one noticed it. This is the best scientific discoveries sometimes are those things which you can't see it even though it's under your nose. So I strongly recommend, and there was a beautiful theoretical paper published back to back with the experimental paper that showed how that worked. If you Google nature angelfish stripes, you'll pull up the reference. Okay, so let's talk about implementing chemical fields. We've already seen in Wizard that there is a dialog box called chemical fields, which we've skipped so far in this class. Now Connor and Andrew have not been skipping it now, but we've skipped it so far. And Wizard will let you build chemical fields. And I'll walk you through it, and then we're probably going to run out of time and not have time to do the exercise. But I'll give you, for homework, some simple exercises on chemical fields to do. It may be a day or two to get the homeworks ready because I didn't know where we'd get to. So let's just walk through the process. And maybe I should do this live while we're doing it. Sorry about this. Let me rearrange my screen, and then I'll do it live. Reshare. So I'm going to start a new simulation. I'm not probably going to finish the simulation today, but let's just call the simulation diffusion. I'm not going to worry too much about my cellular initial conditions. I'll create a cell type because I have to have at least one cell type or I may have a problem. And now I come to this chemical fields. Can people see the wizard? So I now have this chemical field box that I've been ignoring. And it says chemical fields diffusants. And some of these things may have changed slightly in their display because some of these screenshots are from older versions of Compusal, so I apologize. And so now I'm going to type in the field name. I'll call it oxygen, just for the sake of having something. And I hit add. Notice I have an option, diffusion solver FE, flex, fast, kernel, and steady state. Winds up there is a reaction diffusion solver, but I have to type that one in by hand. It's not offered there. Don't know why. Typically, we'll just use diffusion solver FE. There's also this button, enable multiple calls of PDE solvers. The numerical methods that we use are not absolutely stable. And so occasionally you have to tell the system that it needs to use a higher resolution than it would do by default. That's what that button is for. In the old days, you had to do all of that by hand. Now it does it pretty well. So you should be okay with it. So we'll hit add. Field labeled contains white space. Did I manage to? Clear table. Let me try it again. I must have hit a space in my name when I was talking. Yeah, there. Okay. And I can create multiple fields. Not dramatic. And we're going to stick with diffusion solver FE. We've added it. And suppose I wanted to have another field. I'll call it glucose, just for the sake of having something else. Glucose, add. That's it. Now I hit next. And I'm going to skip all of this, hit finish. And what this will do is generate XML XML, and there's going to be a steppable, which is not one we've seen before, which is going to be called diffusion. And the first line of the steppable will be steppable type, in this case, diffusion solver FE, which is the one we chose. Inside of diffusion solver FE, there will be one or more chemical fields, and they're all going to have the same basic structure, and there are a lot of options here. And we can walk through those options a little bit, one at a time. The first thing that you're going to see is diffusion field name equals oxygen. And then later on, you're going to see field name oxygen again. And so there are going to be a couple of places where you may see the same information multiple times. And I do not guarantee what will happen if you make those not agree with each other. So I recommend leaving them the same. You're then given the question, what is the diffusion constant? Because that's the key thing for diffusion. And there'll be something called global diffusion constant. And that sets that number there. We haven't talked about decay yet. We'll talk about that either later in the last 5 minutes of the class or next week. And there are going to be some additional options. Initial concentration. And I think Connor and Drew were talking about using things like the initial concentration. You can write simple algebraic and functional forms of x and y and z, the positions. So if you want to create a linear gradient or a circular gradient, you can do that. You can't do very elaborate math, but you can do basic math. You can read in the concentration from a file. It reads it in ASCII, so those are big files typically, but you can do that. And that defines the basics of the field specification. You can also create these things in Python, or at least the values in Python if you prefer. That tends to be slow, but if you're initializing it once, it's not the end of the world. Any questions about that so far? Does that make sense? The next thing that you have to think about is boundary conditions. Our fields and our cells represent a region of space, and it's not the whole universe. And so we have to decide what happens at the boundaries. And for the cells, we sometimes have used fixed boundary conditions, no flux boundary conditions. Sometimes we've used periodic boundary conditions. For chemicals, this is also going to be important. We have to ask the question, what happens at the boundary? For a cell, basically there are two options. Either the cell can't cross the boundary, it crosses the boundary and goes to the other side of the lattice, periodic boundary conditions. There are more options that are possible for chemical fields. Typically, we're going to assume periodic boundary conditions. So if I think of a molecule exiting one side of the lattice, it comes back on the other side. And that's something that we're going to do often. On the other hand, if I imagine that my lattice represents a volume of liquid inside a test tube, the test tube has a rigid wall. And the chemical doesn't cross the wall. And in that case, I would have no flux boundary conditions. The chemical can't cross the boundary. The other thing that happens, and this is a little bit different from what happens with cells, is that I might have constant value boundary conditions. And a simple way of thinking about this would be for temperature. Suppose that I bring an object into contact with a heater, and the heater is at constant temperature. I'm going to have heat, energy, pumped into my object from that heater. And the temperature of the heater is fixed. And so I could have a constant value boundary condition where the chemical field is fixed to have a constant value at a surface. I could also have a constant flux. Imagine I'm pouring water into my system and I pour it in at a constant rate. I sprinkle ink into my system at a constant rate. And so I have the possibility of periodic boundary conditions, no flux boundary conditions, constant flux boundary conditions, or constant value boundary conditions. And each one of those has somewhat different mathematical properties. Constant value boundary conditions are nice because the concentration of the chemical can't go above that constant value. So it's bounded. Constant flux, in principle, if I keep pouring ink in, the amount of ink can go to infinity. And so the amount of ink I'm going to get is going to depend on the rate at which I add it and the rate at which I take it away. And I'm going to have to decide on a biological basis which of these makes sense. And it's going to vary depending on the situation. Typically, if I have something like oxygen coming from blood vessels, I can treat the concentration of oxygen at the surface of the blood vessels constant. So it'd be constant value boundary condition. In other cases, I'll have constant flux or no flux boundary conditions. And so there are going to be a series of choices. And if I look here in lines 56 and 57, I have constant value and plane position is minimum and maximum, and I have the x direction, the y direction, the z direction. I can have constant flux or periodic boundary conditions. And so I'm going to have to make choices on all of those in each direction. And so that looks like this. And each one of those is going to have quite big differences in the results. And so I guess we're going to be out of time. I was hoping we would do the first exercise, which is to do diffusion with different boundary conditions. Let me see if this will run. I haven't tried it. I wasn't actually planning on running it. Let me see if this will run. And if it gives us something. If it crashes, we'll say that that's the end of the class. Well, it didn't crash. The cells are not important in this. Let me look at glucose. Here's a glucose field. The boundary conditions here are a little bit strange. I have here at the top a constant source, here at the bottom a constant removal, a constant negative flux. The concentration's gone negative, which it shouldn't. My cells are interfering with, are acting as a source of material. And so this is a random simulation showing the changes in chemical concentration for a particular set of diffusion equations. And again, the defaults here for the way this is set up are not necessarily the greatest. And so I'd like you, and I'll assign this now as homework since we didn't get to it. I'm going to assign to you some simple exercises to begin to explore how to set up a chemical concentration, what happens when you have diffusion, what different boundary conditions do. And I hope that that will begin to give you a sense of how these things behave. And I'm sorry if we ran out of time, but I did want to at least get through the basics of chemical fields. And then next time we'll come back and we'll work in more detail with chemical fields. And once we have chemical fields working, we're going to have to ask the question, how do they interact with cells? And we're going to find that there are a couple of things that happen. One, cells can move up and down gradients of chemicals through chemotaxis. Two, cells can absorb and secrete chemicals. Three, the chemicals can make the cells do things. They can be the nutrient that allows the cell to grow. They could be a toxin that kills the cancer cell. They could be a signal that tells the cell to differentiate. And so cells can interact with chemical fields in a variety of ways. And one of the beauties of emergence in biology is that the cells are continually responding to the chemicals in their environment, but they're also changing them. In particular, cells will do things like they will secrete a signaling molecule, and they will respond to the molecule they're secreting themselves. And so it's not that there's a background of chemical that the cells are responding to, and that those are separate. The cells are creating the environment that they're responding to. And that allows for the beauty and complexity of biological organization. A big part of the beauty of biological organization comes from the fact that cells are creating an environment to which they respond. And so you can't really separate the environment from the cell, because the cells themselves are creating that environment. All right, so you've been very patient. Is there any questions before we break for tonight?
I just had kind of a administrative one. I don't know how everyone else feels, but I think it would be a lot easier if you could make just the weekly homework assignments due on Wednesdays, because I feel like at least I feel like I work on the homework and then I have all these questions and then they're usually answered in lecture, but then they're due like 4 hours later, and that's kind of difficult. So I think it might be a little easier if it's just due the next day. But
okay, there are
it's
two
up to you.
things to say. Two things to say. If there's a problem that first, I can't promise that I always respond to emails on time, but I do try to respond to emails over the weekend. So if you're doing the homework on Saturday and you ask me a question, I try to get you an answer. Sometimes my life is complicated or I don't see the email, but I'll try to answer your questions over the weekend. And the reason that they're typically do weekdays on the class day is that in my experience, most people do the homework over the weekend. But some people, of course, try to do it the day before, which is fine, or during the day. That's all right, too. The second thing is, as I've said before, if there's a problem that you feel we didn't cover that you need to see in class, you're always free to write in the homework, I'm holding this over till next week, and then hand it in a week later. So, of course, if you want to get it out of your hair and hand it in on Wednesday, that's fine. The only reason that I typically want them have the assignment due for the class is that I hope that people, if I make it due tomorrow, then my guess is that not everybody has the discipline to try it before class. And my hope with these problems sometimes is that people will try it. And that way, when we come to class, you can ask the question, I tried this, it didn't work, or I don't understand how this works. The kind of questions you've been asking are excellent questions. But I really like to have those come up in the class so that I can answer them for everybody. And so that's the motivation. But if you prefer to hand in the homework sort of for yourself on Wednesdays rather than Tuesdays, I can live with that, because typically I don't get to grade the homeworks until the weekend, because I'm also busy during the week. And so I would prefer, I mean, if there's just, we can have a vote, people. This is fine. Again, you have the freedom. I treat you as people who are adults, and I don't mean that in a negative way at all, I hope. And so if you tell me that you need more time to do the problems, I'm not going to protest. And again, but do people think it would be helpful to have the official due dates a day later? Or is it something that we're not exactly a democracy, but I certainly don't want to make people miserable. So you tell me, is how people feel.
I personally, typically what I end up doing is that I try to solve the assignments and then I try to go back to them for a few hours after class. There's been a couple of times where I've still been stuck at the end. I don't know if handing it in the morning would make an endless difference, but I guess it would give a few more hours. Personally, I will still always try to have them done before class just because I find that that's the only way that I can make sense of where I would get stuck anyway.
I mean, I think again, Josh, and I'm not trying to dismiss your concern, but my real suggestion is if there's a problem where you feel it needs to be handed in Wednesday, hand it in when you're ready. But the following week is all right. In other words, put in the homework. I didn't get it done. And as I say, I typically don't look at the homeworks until Friday or Saturday. And so you have some wiggle room. If you hand it in Wednesday instead of Tuesday, I'm not going to know the difference. So I guess the answer is you've already got what you asked for. Is that okay? But
Yeah,
please
I was
try
just
to work
wondering. I
out.
didn't know.
Yeah. I'm not. I mean, I say that late homeworks will be penalized, but in fact, what I mean by that is don't hand in the homeworks 3 weeks late. If you need an extra day or two, it's okay.
Yeah, that's mostly what I was wondering.
Okay.
Thank you.
And I'm sorry if I made a big thing out of that, but I guess it was a philosophical question, so I answered it like a philosophical question. Any other concerns or comments? I understand this course can be problematic in all sorts of ways, and I do appreciate your patience.
I have a general request, and also if it ends up that no one else is having issues with this, I can just talk to you about separately. But a couple weeks ago, there was a homework assignment for iterating over cell neighbors that I had a little bit of trouble with, and that came up again this week. And I feel like I made it further this week, but my code still didn't work. So if at some point we could, if it's just me, then I'm happy to just do it in office hours at some point. But I just wanted to throw out there that I had gotten stuck there once or twice.
Okay, so that's a good question. So I will tell you something, which is I had a set of slides which I think were distributed, which went over that. And I had planned on doing that last week in detail, and we ran out of time. And when I prepped the lecture for this week, I had the choice, which was I going to get us into chemical fields and move us on to the next thing, or was I going to take half of the lecture to do iterators over neighbors? And I was torn because I understood the issue that you're raising. And I also knew that people wanted to see chemical fields and get started on that. And I'm happy to go back. I have the deck exists. It's actually in the deck here that's uploaded. If you want to look at it. And so if people want me to spend a half an hour going over iterators for neighbors next time, I'm happy to do that. If there isn't a strong groundswell of needing that, then I'm happy to sit with you one-on-one or set up an office hour where I say I'll do that, and then anybody who wants to join it can join. How do people feel about that?
Or even if I was about to say, even if we had like a group that could get together and do some of the homeworks together, maybe it's a good idea to just bounce ideas off and see problems of other people. Not encourage you to share the results, but trying to help each other.
I don't mind if you work together on I do want you to learn. And so the one thing I would ask you to do is not have a person X who's done it before do it and then everybody copy it. But if you collaboratively do it and you say, I worked with so-and-so, suppose makes sense. For example, Connor, you and Drew are working together. You're working on a project, so you're going to be solving problems together. If you say that Connor and Drew, in your homework, you say, I worked with Drew on this, and we wrote it up separately, but we both worked on developing these together. I'm okay with that. That's not, as long as it's acknowledged, it's fine. My only concern is I don't want people to not learn the material. And again, I think it's hard to remember this, but I do believe that the purpose of these homework assignments is to help people learn. It's not to torture you. You may feel that it's to torture you, but it's not. And so if you're stuck, please reach out to Giuliano. Reach out to Joel's done some of these things before. Reach out to TJ. Reach out to each other. I think that if you have a study group and you work on these problems together and you come up with collective solutions, that's great. Because as I've said repeatedly, everyone in this class comes with a different background and you can learn from each other. And I promise you that I spend a lot of my time helping people who are professors do this kind of work. I also learn from them. And very often I will sit down with somebody comes to me with a problem and we start working on it. I didn't know that answer, or I had to come up with a new way of solving it. And so I don't certainly know everything about these kinds of things. I know some things, and I have a particular way of trying to teach it based on my experience of how people learn in the past. But there's a lot of variety to that too. And I recognize that the approach that I take here may not be what people need directly. And so learning from each other, I think, is great. So feel free to work together. The only thing I ask is if you work together, let me know. And just as I say, it's fine if you try the problem and then work on it more after class. But if I do the problem completely in class and you hadn't worked on it before, then my concern is not you'll get a grade for something you didn't do. I don't care about grades. But my concern is that if you copy what I did in class and you didn't try it, then the next time you have to do it yourself, you may not know how to do it. And my goal is to try to help you learn how to do these things yourself. And I'm very open to suggestions about ways to make that more effective. And also recognizing that different people learn in different ways. But feel free to work together. Just let me know. I mean, not that I won't notice, but let me know anyway. Okay. Is that all right? Anything else about logistics? I realize it's a bit of an adventure every time. And it's different every year. What we cover, the rate at which we cover things, what people want to do, always different. Compusal changes a little bit, but and biology doesn't change. But the way we approach it changes. So I hope that addresses some of these concerns. And don't be intimidated about homeworks too much. And do feel free to ask each other. And I realize when it's virtual, you're not interacting in person as much, but we seem to have pretty good conversations here. People seem to be comfortable asking questions. And Giuliano, thank you for being here. We seem to be pretty comfortable asking questions and talking. And you see, you can tell me I did something wrong or make a suggestion. I don't bite. My cat bites, but I don't. So, and if you don't want to do it in public, you so we can set up meetings and talk about things too. All right.